Good morning children, I am Sarita. Now I am going to teach you infertility. Infertility means inability to produce children in spite of unprotected sexual cohabitation. So let's see, infertility means inability to produce children. So the reason for inability may be physiological, congenital diseases, drugs, immunological or even psychological. Nowadays, the infertility clinics are available to diagnose the disorder of infertile couple and also provide correct treatment to have children. The infertile couple could be assisted to have children through special techniques called art. Art means assisted reproductive technologies. Assisted reproductive technologies. So through the special technique, the infertile couple could be assisted to have children. Is it clear? And now we can see what are the different types of art or assisted reproductive technologies. The types of art, the first one is test tube baby program. Test tube baby program. And the second one is gift. So gift means gamete intra Fallopian transfer. So that is gift. And the third art is ICSI, that is Intra Cytoplasmic sperm injection. And the fourth art is AI, that is artificial insemination. Artificial insemination. So these are the different types of art, assisted reproductive technologies. Okay, and we can see the first one, test tube baby program in detail. So test tube baby programs. So in test tube baby program, actually this is a misnomer or misname. It does not mean that the baby is developed inside the test tube. Actually, fertilization takes place in the laboratory under sterilized condition or in vitro condition. And in this method, we can see what is happening here. The ova from the female are selected or collected. So the ova from the female may be wife or donor, and sperm from the male can be collected. The male may be husband or a healthy donor. Okay, so these ova and sperm are induced to form a zygote by the process of fertilization. So this fertilization occurs in the laboratory. That's why this technique is known as IVF, that is in vitro fertilization. In vitro fertilization. Okay, so in vitro fertilization means the fertilization or the formation of zygote occur in the laboratory under in vitro condition or sterilized condition, and that fertilization is called IVF. Okay, so this IVF is followed by CIFT and IUT. Let's see what is meant by CIFT and what is meant by IUT. Okay, so here, as I told you, zygote is formed under in vitro condition when the zygote attain up to 8 blastomere stage. 8 blastomere stage. It can be directly deposited into the fallopian tube. Fallopian tube of the female. Okay. So this process of deposition of 8 blastomere embryo stage into the fallopian tube of the female body is called CIFT. So CIFT, what is the full form of CIFT? Zygote intra fallopian transfer. Fallopian transfer. Okay. And now we can see, so this zygote, when it attains more than 8 blastomere stage, it can be directly deposited into the uterus. So when the zygote is having more than 8 blastomere stage, more than 8 blastomere, so it can be directly deposited into the uterus. And that process is called IUT, that is Intra Uterine Transfer. Is it clear? So IVF is followed by two techniques, CIFT and IUT. CIFT is the zygote intra-fallopian transfer. IUT is the intra-uterine transfer. So when the zygote or when the embryo attains up to 8 blastomere stage, we can directly deposit the embryo into the fallopian tube of the female body through the technique CIFT. When the embryo is having more than 8 blastomere, it can be directly deposited into the uterus through the technique IUT. Is it clear about test tube baby? Okay. Next we can see the second one. Before that I will show you one video. So here you can see the ovaries consist of ova. The ova can be collected under sterilized condition or in vitro condition. And the sperm can also collect from the male and it injected into the ovum. So normal fertilization occurs in the laboratory under sterilized condition. So zygote will be formed. And zygote, when attain 8 blastomere stage or more than 8 blastomere stage, it can be directly deposited into the fallopian tube or inside the uterus. So after the deposition, it can normally grow and develop into fetus and then into baby. Okay. Is it clear? Now we can see the second method, GIFT. GIFT, what is the full form of GIFT? Gamete intra-fallopian transfer. So from the name you can see gamete is transferring here, not zygote. If it is zygote transfer, it is called the CIFT. Okay, so gamete transfer, if a female or a wife is having the problem of irregular ovulation or if she is not capable to produce ovum properly, this method, GIFT, can be done. And here in this method, the ovum can be transferred into the female from a female donor. So the ovum or the gamete can be transferred into the fallopian tube of the recipient female. Okay. So here transfer of gamete. Gamete means ovum into the fallopian tube. Fallopian tube. So this ovum or the gamete can be collected from the donor female. Okay. And here, after the deposition of this gamete into the female fallopian tube, normal fertilization will occur after copulation. Is it clear? So this is GIFT. So, do you know the difference between CIFT and GIFT? CIFT is the zygote intra-fallopian transfer. GIFT is the gamete intra-fallopian transfer. It's clear. Now we can see the third one, ICSI, that is intracytoplasmic sperm injection. This also can be done in the laboratory. So, this is the formation of embryo in the laboratory. In the laboratory, by injecting the sperm into the ovum. It also done in the laboratory under sterilized condition. Okay, next we can see the fourth one. Fourth one is artificial insemination. Artificial insemination. So, in this method of art, when a male is suffering from low sperm count or is not having sperm motility, proper sperm motility, this technique, artificial insemination, can be done to have children. Here in this method, the sperm can collect from the husband or from the healthy donor, and it can directly inject it into the vagina or into the uterus. So this is called the artificial insemination. So here the sperm can inject into the vagina or uterus directly. First, sperm need to collect from the husband or from the healthy donor. Then the sperm can directly inject into the vagina or uterus. Is it clear about infertility and art methods? Hope you understood everything about infertility and different methods of art. That's all for today.